have to discuss the next part of the lesson under heredity, mutation. What is mutation? Let's see. If you have a gene, a dominant gene A in a particular locus, in a point of chromosome, the gene may get changed into small a. So this is called mutation. And such mutations will be transferred to the next generation. So what is mutation? A sudden, inheritable change that occurs in a gene or in the chromosome. Suppose you are taking a chromosome. This is a long chromosome. Maybe a part of the chromosome is deleted, or a part of chromosome is added, or it is being inverted, or it is being translocated. So anyway, either change in gene or change in the structure of chromosomes or in the number of chromosomes, that is called what is known as mutation. A sudden heritable change in the structure of a gene or in the structure of chromosome or in the number of chromosomes. That is called mutation. All of a sudden changes occur. So that what is happening? The external phenotypic expression is also changed. The external visible appearance is also changed because of sudden change. Now, who coined the term mutation? Now, the theory of mutation was proposed by a Dutch scientist, Hugo de Vries. 1901. So he only coined the term mutation and also proposed the theory of mutation. While he observed in a plant some phenotypic changes, external visible changes, the name of the plant in which he observed mutation, a sudden change which is being actually inherited, was Enothera lamarckiana. This is the name of the plant where he observes some phenotypic changes all of a sudden in a population. Now the common name for this plant, evening primrose. That's the name of the plant, the common name. So he observed mutation that one. Accordingly, he proposed a mutation theory and also coined the term mutation. I mentioned already, what is mutation? Sudden change that occurs either in a gene, change in structure of gene, or change in structure of chromosome. So either change in structure of gene or change in structure of chromosome or change in number of chromosomes. So mutation occurs at three levels, either in the genes or in the structure of chromosomes or in the number of, that is, chromosomes. Now we have to go for the first one. So what are the three types of mutations? What I mentioned here. One, change in structure of gene, that is called gene mutation. Change in structure of gene, or change in chromosomes, that is called chromosomal mutations. Now in chromosomal mutations, it occurs either due to change in structure of chromosomes, or change in number of chromosomes. So these are the three major types of mutations, either change in gene or chromosomal changes. The chromosomal mutations are of two types, either change in structure of chromosomes or change in number of chromosomes. Now the gene mutation is also called as point mutation. The another name, point mutation, as it affects a particular point in a chromosome, namely the gene. Now I will take the first one, the chromosomal mutations. I mentioned already, chromosomal mutations may be due to change in structure of chromosomes or change in number of chromosomes. I will take what we mean change in structure of chromosomes. So, some visible structural changes occur in the chromosome, and that occurs due to error in cell division. As a result, we have change in number or arrangement of genes. So, change in structure of chromosomes results in change in number and arrangement of genes. It is due to error in cell divisions. Now, what are the different methods by which change in structure of chromosomes occur? It may be because of deletion of a chromosome. If you are taking a chromosome, a particular chromosome, a part of the chromosome is deleted, or it may be due to what is called addition. For example, in a particular chromosome, a piece of chromosome is added. So a piece of chromosome is added. This is what is called actually addition. As a result, we have a particular part of the chromosome is being represented twice, and this is also called, instead of this one, duplication. In some cases, we have what is called translocation. So these are all the methods by which change in structure of chromosomes occur. Either deletion, a part of the chromosome is deleted, or duplication, particular sequence of genes being present twice. For example, represented here 1, 2, 3, here also 1, 2, 3. Duplication. The arrangement of genes normally found twice. That is called duplication. Translocation. From one homologous member, suppose this is one chromosome, a part of the chromosome is deleted and that is added to another homologous member. You see, this is added here. This is what is called translocation. Here exchange occurs between the homologous members. Here exchange occurs between non-homologous members. Maybe this is the first chromosome, it may be the fourth chromosome. A bit of chromosome is deleted from the first and added to, that is a fourth. This is called translocation. Deletion, removal. Duplication, addition, that results in duplication of a particular series or sequence of genes. Translocation, a piece of chromosome is deleted from one chromosome and added to another homologous chromosome. This is called translocation. Another method, what we call inversion. Now here, suppose you are taking a particular chromosome, I am taking that one, the sequence of genes, 1, 2, 3, 4, 5, 6, 7. Now the chromosome is undergoing what is called inversion towards 180 degree, inverted. As a result, the sequence being changed, the arrangement of genes being inverted. So 1, 2, that is what we have 5, 6, 7, 3, 4. See, the arrangement of genes being normally changed. This is because of the inversion, the rotation of the chromosome towards 180 degree. These are the methods you will be studying in higher classes. Anyway, change in structure of chromosomes occur during cell division, either because of deletion of chromosome or duplication of chromosome or translocation of chromosome or inversion of chromosome. So that the change in number and arrangement of genes occur because of these activities. Only change in structure in a particular chromosome. Now let's go for change in number of chromosomes in the next part. Now, what is happening? Now the next part of mutation is mutation due to change in number of chromosomes. When there is a change from a normal diploid set, the phenomenon is called ploidy. Change from a normal diploid set is called ploidy. So, deletion or addition in the number of chromosomes in a cell is called ploidy. Deletion or addition in the number of chromosomes in a cell is called ploidy. So it may be the addition of entire set of chromosomes or the addition of individual chromosomes or loss of individual chromosomes. So accordingly, the ploidy can be divided into two types. One, euploid, another one, aneuploid. So ploidy, variation in normal diploid number of chromosomes. So what is diploid? Here addition of entire set of chromosomes occur. Suppose you are having, for example, this is a normal diploid set. Here one set is added, so we are getting 3n. Or two sets are added, so that we are getting 4n, etc. Or three sets are added, so that we have 5n. The maximum number, you see that one, that is two sets. To these two sets, four sets are added, so 6n. According to the sex of chromosomes, it's called triploidy. It's called triploid. And this one, tetraploidy. Tetraploidy, based on the sex. And this one, pentaploidy. This one, pentaploidy. And this one, hexaploidy. Hexaploidy. Now, this is called AP. So, more than usual number of chromosomes. The organisms normally bear more than usual number of chromosomes. Now, instead of 2n, they have either 3n or 4n, 5n or 6n. This condition is called diploid. Now, in the case of triploidy, the organisms are called triploid. They have three sets of chromosomes. Triploidy condition in animals and plants cause the animals and plants sterile. Cause the animals and plants sterile. For example, triploid tomato, triploid watermelon. Now, in the case of tomato, we have 24 chromosomes. It is equal to 2n, tomato. Or in the case of, for example, paddy, wheat or paddy, I am taking the 24, that is 2n, that is paddy. Like that. Even if we are taking wheat, we have 2n is equal to just actually 14 chromosomes. Now, in the case of maize, we have 2n is equal to 20 chromosomes. Now, we have some examples given in your book. Now, the triploidy results in triploid organisms in animals and plants which cause sterility. But about the tetraploids, there are more advantages because they produce increase in size in fruits and flowers. So they are valuable in what is called horticulture or agriculture, production of just large sized fruit, large sized flowers or large sized vegetables. So it is advantageous when compared to triploid. So in a book given an example of triploid. Two examples given, one animal and one plant. Triploid banana. Triploid banana. Triploid banana. Now, in the case of diploid banana, so it has normally 22 chromosomes, that is equal to 2n. But in the case of triploid banana, as n is equal to 11, now it has actually 3 sets of haploid chromosomes, so 33 chromosomes. In triploid banana, we have 33 chromosomes. In the case of diploid banana, we have 22 chromosomes. So the scientific name of the banana normally Musa paradisiaca. This is a scientific name of banana. So triploid banana normally larger in size, seedless. No seeds are found in the case of triploid banana. Now the diploid banana with the 22 chromosomes normally smaller in size and seeded appearance with the seeds. This is without seeds, seedless banana. This is banana with the seeds. Now you see that one here, the diploid number is 22 and the haploid number is equal to 11. So total number of chromosomes in triploid banana is 33. That causes what's called gigantism, more growth and also seedless actually, that is fruit. And likewise also we have received what is called watermelon, like that. So many individuals have produced triploid tomato, etc. Now, this is an example for plant. There is also another example for triploid organism. That is an animal. There is an oyster. Oyster. So, Crassostriga gygos. This is an oyster. This is a Pacific oyster living in Pacific Ocean. Now the diploid oyster has 20 chromosomes. That is, n is equal to 20. Sorry, n is equal to 10. The diploid number 20, n is equal to 10 chromosomes. But in the case of triploid oyster, we have 30 chromosomes. As n is equal to 10, we have 10 into 3, 30 chromosomes. Now this oyster is large in size, more fleshed inside when compared to, there is a normal diploid oyster. So one example for plant, one example for animal is given in the syllabus. I will represent the photographs in the video. You can just compare that one. So what is the difference between these two? So the number of chromosomes here 20 and then 3 just 30, and that causes more growth. So it contains more flesh. That is, its visceral mass, the weight inside is more when compared to what's called the normal diploid oyster. So this is about diploid, the examples. The entire set of chromosomes is added. So they have more than usual numbers. Triploid organisms are sterile. Tetraploid organisms are more advantageous because they cause increase in size of what is called the fruit and flower. Now what is aneuploid? What is aneuploid? So what is aneuploidy? The next type of change in number of chromosomal mutation. Now it may be a gain or loss of chromosome. In the normal diploid set, gain or loss of chromosome in a normal diploid set. This is called aneuploid. For example, gain. One individual chromosome is added to a diploid set. 2 chromosomes are added to a diploid set. 1 chromosome is deleted from a normal diploid set, or 2 chromosomes are deleted at a maximum from a normal diploid set. This is called aneuploid, either gain or loss. When 1 chromosome is added to a particular pair, so instead of having 2 copies, we have 3 copies. Suppose we are taking the 21st chromosome, 21st. In a normal individual, we have 2. A pair has 2 chromosomes. But in the case of 21 trisomy, this is called 21 trisomy, where the 21st chromosome is found in 3 copies instead of normal 2. That means 1 chromosome is added. As we have 3 copies of one particular pair, it is called trisomy. In the case of tetrasomy, 2 chromosomes are added to a particular pair. So we have 4 copies are present in a particular pair instead of 2. Now monosomy, 1 chromosome is being lost. Out of just 2 members, 1 chromosome is lost, and this is called monosomy. In a pair, only 1 member is present. Now the last one, nullisomy, 1 particular pair is completely lost. I represented a dotted line. That means both the chromosomes in a pair being lost. This is called nullisomy. Now in your syllabus given, what example? For trisomy, that too 21 trisomy. What is called Down syndrome? One example is given for trisomy. What is called Down syndrome? This is the name of the disease, genetic disease. So syndrome means signs and symptoms that characterize a particular abnormality in a genetic disorder. Down syndrome. This Down syndrome was first reported by Langdon Down, hence the name Down syndrome, in 1866. He only observed the syndrome. So the Down syndrome is an example for 21 trisomy. It is an example for 21 trisomy where we have 2n plus 1 number. A particular pair, what is called the 21st pair. This is the 21st pair of chromosome. The 21st pair of chromosome is formed in 3 copies instead of just 2. You see, 3 copies. That's why it's called 21 trisomy. This is because of actually some error during celebration. So Down syndrome is an example for 21 trisomy where we have 2n plus 1 condition and example for unemployed. Now what are the symptoms? In all genetic disorders we have mental retardation. The symptoms of Down syndrome are: number 1, mental retardation. Mental retardation. Number 2, delayed development. Delayed development. This is another one. Then weak muscle tone. Weak muscle tone. That is a muscle contraction. Muscle tone means nothing but the muscle contraction. Then we have normally some behavioral problems. Behavioral problems. And vision and hearing affected. Disability. Vision and hearing disability. The tongue is protruding out. The mouth is kept constantly open. The space between the eyes is longer. Flattened nose, etc. Only 5 symptoms are given in the book. The mouth is always kept open. The tongue is protruding out. The space between the 2 eyes is longer. These are all some more symptoms for Down syndrome. So I have given a beautiful picture, an affected male, along with the karyotype of that is what is called the Down syndrome. In the video you can see later. So that's about what we have aneuploidy Down syndrome. So far we have studied mutations are caused due to change in structure of chromosomes and change in number of chromosomes. Now the next one, point or gene mutation. What is point or gene mutation? Why is it called point? As it affects a particular point in a chromosome, that is a gene, it is called point mutation. As it causes normally changes in a nucleotide sequence. Nucleotide sequence means you see that one in a gene we have, for example, these are the bases, nucleotides, like this. When you have a change in a nucleotide sequence, then it is called point or gene mutation. This is the original sequence. Sometimes a base has been removed or a base has been added. Like that we have change in the structure of gene or DNA occurs, and that is called actually point. Changes in a nucleotide sequence of a gene. So the bases are arranged, the nucleotides are arranged in a specific sequence in a DNA like this. When there is a change in the arrangement or sequence of genes, that results in mutation. What are the causes? It may be due to substitution. It is called substitution gene mutation. One base has been replaced by another base. For example, I am taking here AGC sequence. This is called nucleotide sequence in DNA. Now, adenine has been replaced by what is called, you see that one, uracil. A has been substituted by U, then it's called substitution. The best example for this one is disease, sickle cell anemia. I'll tell you more about this one in a few seconds. Now, deletion. Suppose this is the nucleotide sequence in a particular gene. Now, what is happening? This gene has been deleted. That is called deleted gene mutation. Or a base has been added. You see that one, adenine base has been added to this sequence. You see that one, A is added. So the entire frame has been changed. That is called insertion. Like that also we have inversion, change in sequence of actually bases in a reverse order, and that is what is called inversion of one or more than one nitrogenous basis. So substitution of one or more than one nitrogenous basis, deletion of one or more than one nitrogenous basis, insertion of one or more than one nitrogenous basis, or inversion of one or more than one nitrogenous basis, main cause for the point on gene mutation. In all gene mutation, it results in the production of abnormal non-functional protein of an organism, because normally the genes code for proteins. When the gene is defective, an abnormal protein, non-functional protein will be produced. I'll tell you one example. Say an example of the substitution mutation is sickle cell anemia. What is sickle cell anemia? It is an example for sickles, actually a mutation, a substitution mutation. Now, in a hemoglobin, the respiratory pigment, the red pigment present in the RBC is formed of two components. One, heme, another one, globin. The globin is a protein part. Globin is a protein part having 4 chains, 2 alpha chains and 2 beta chains. Now, in the beta chains, at the 6th amino acid, at the 6th place, normally the beta chains at the 6th place contain one amino acid, what is called glutamic acid. One amino acid called glutamic acid. This is amino acid present in the 6th place or the 6th position of beta chain. Now this glutamic acid is coded by a nucleotide sequence GAG. This is for glutamic acid. This is the code, genetic code for glutamic acid. But when this GAG has been substituted, for example, A has been substituted by U. When A has been substituted by U, what will happen? The change in the genetic code occurs. This code, the genetic code, is normally meant for valine amino acid. Valine amino acid. Valine amino acid. So, here substitution occurs at the what is called gene level. A has been substituted or replaced by uracil. So instead of glutamic acid at the 6th place in the beta chain, we have valine amino acid. That causes the production of abnormal hemoglobin. That is, alteration in the gene causes the production of abnormal part of protein, that is a globin. And the cell having abnormal hemoglobin becomes sickle shaped. The cells having, the RBCs having abnormal hemoglobin have sickle shape at low oxygen tension, hence called sickle cell anemia. Normally the RBCs are circular and biconcave, but in the case of sickle cell anemia, it is like a sickle, what we have the sickle, the knife used for harvesting the field. So it becomes sickle shaped, and that's why it's called as sickle cell anemia, a disease caused because of abnormal production of the protein, that is a globin, due to alteration in a gene. Here it is an example for substitution mutation, the gene mutation. A base adenine has been replaced by uracil. So this is the last part of the lesson. So I concluded this part. Go through the videos and study well. I pass on to the next lesson in the next class. Thank you.